So here goes part 5 of our lecture on photosynthesis in chapter 10, and our last part. So we left off with beginning our look at the Calvin cycle, and I wanted you to know that the Calvin cycle uses the chemical energy of ATP and NADPH produced through those light reactions in the electron transport chains in the thylakoid memory. These molecules are now used to reduce carbon dioxide to sugar. So when we're looking at the Calvin cycle, carbon is going to enter the cycle as carbon dioxide, and carbon's going to leave the Calvin cycle as a sugar named glyceraldehyde 3-phosphate, which we shorten to G3P. For a net synthesis of one molecule of G3P, the cycle has to take place 3 times. So we have a circle of reactions that are occurring in the Calvin cycle, and that cycle, that turn, has to take place 3 times in order to make just one molecule of glyceraldehyde 3-phosphate. So that means we also need 3 molecules of carbon dioxide entering the Calvin cycle in order to produce that one G3P. So the Calvin cycle can be broken into 3 phases. The first phase is the introduction of carbon dioxide, which means we're going to have carbon fixation occurring. And this fixation process is occurring through the enzyme Rubisco, which we shorten to be RUBP. So carbon fixation is our first step. Our second step is a series of reduction reactions. And in that reduction reaction, we actually get to our production of the G3P. And then the last phase, the third phase of the Calvin cycle, is the regeneration of the CO2 acceptor, which is RUBP, or the Rubisco. And also, and remember, regeneration of an enzyme simply means that the enzyme has been freed up to do its job again. The other part of this regeneration phase is also, as we are using the ATP and we're using the NADPH molecules throughout the Calvin cycle, what's occurring is that they are returning or regenerating back to their NADP plus form and ADP form, which means they are now ready to be electron acceptors and the objects of phosphorylation in the light reactions back in the thylakoid membrane. So some facts for the journey from carbon dioxide to a glucose molecule. First of all, carbon dioxide has very little chemical energy. It's already fully oxidized. Whereas our glucose molecule, our C6H12O6, contains a lot of chemical energy. It's highly reduced. So in order to go from that CO2, that fully oxidized, to that C6H12O6, fully or highly reduced, we have to go through an endergonic process. So the synthesis of our molecules is going to require the input of a lot of energy. And so what happens in the Calvin cycle is we end up with a series of many small but uphill steps, meaning they require energy in order for them to proceed. So that means in order to lower the activation energy for each of those steps, we're going to need the presence of a number of specific enzymes. And also we're going to need the energy that we have stored in the ATP and the NADPH from the light reaction. So just to go over this very quickly, you don't again need to memorize every molecule in this cycle. There are just a few that I want you to know. So what we start with in phase 1 is the fixing of carbon, or the fixation of carbon. So in the case of what we're doing right now, we're going to actually input 3 carbon dioxide molecules. They actually only enter this cycle one at a time, but we're trying to get our output of 1 G3P, and so in order to do that, we're going to need an input of 3 CO2s. So the CO2 enters the Calvin cycle, and with the help of Rubisco, the RUBP, we're able to fix the carbon into this 3-phosphoglycerate molecule. So we're taking this molecule here, which is a 5-carbon molecule, and we're adding 1 of the carbon dioxides to it in order to make it a 6-carbon molecule. That 6-carbon molecule then quickly splits into 2 3-carbon molecules. So again, we're starting now into our series of reactions that are going to be occurring. So the next phase, after we've fixed the carbon into an organic molecule by adding it to the RUBP, then we are going to go through phase 2 with a series of reduction reactions. And each of these reactions is going to require energy. So we end up needing a total of 6 ATPs in order to move 3 CO2s through the cycle. And we're going to need a total of 6 NADPH through this reduction phase. So 6 of each of these molecules. And at the end of the reduction phase, we end up with our G3P, which can be used. So this is a 3-carbon sugar that can be used to build things like glucose or any other organic compounds. The last phase, 3, is the regeneration of our RUBP molecule. So we want to get back to that 5-carbon molecule. So most of the G3P that's produced is actually going to continue on in the cycle so that it can become the RUBP molecule in order for this cycle to go circling around and around. In order to go from G3P to RUBP, we need the input of ATP again. So in total, we have 6 plus 3, we have 9 ATPs in one cycle, which actually is 3 cycles in this case. So we need 3 ATPs per cycle, and we need 2 NADPHs for every turn of the cycle. So keeping track of the carbon, our carbon accounting here, 3 turns of the Calvin cycle is going to produce 1 G3P molecule, which means we're going to be taking 3 carbon dioxides to produce that 1 G3P. And if we're keeping track of carbons, we can see our G3P is a 3-carbon molecule right here. And so those 3 carbons have to come from somewhere, and they're going to come from 3 carbon dioxides. And each turn that we take through this Calvin cycle is going to take 3 ATPs and 2 NADPHs. Now, if we want to actually make a molecule of glucose, we're going to need 2 3-carbon molecules. So in order to end up with 6-carbon molecule here, we're going to need 2 3-carbon molecules. So that means if we needed 3 turns of the Calvin cycle to make 1 G3P, we're going to need 6 turns of the Calvin cycle in order to get 2 G3Ps, which can then make R1C6H12O6. So again, these carbons in our glucose molecule have to come from someplace, and they're going to come from 6 individual carbon dioxide molecules. Now if we do the math, if we're going through 6 turns of the Calvin cycle, that means we need 6 times 3, which is 18 ATPs, and 6 times 2, or 12 NADPHs, in order to make one glucose molecule. So you can see that is a lot of energy that's going in to that reduction set of reactions to produce something like a glucose molecule. At the end of the reactions, if there is any ATP left over from the light reactions that's not being used by the Calvin cycle, those are going to be used by other parts of the plant cell. So to G3P and beyond, what this is really pointing out is that the 3-carbon G3P, or 3-carbon carbohydrate, is a very energy-rich 3-carbon sugar. And it also is an important intermediate molecule that can go on to make all sorts of different biomolecules. Another actor in our Calvin cycle is this rubisco, which is the enzyme which fixes the carbon from the air and combines it with the ribulose biphosphate carboxylase. So the ribulose biphosphate carboxylase is rubisco, and it's really considered, according to some of our textbooks, the most important enzyme in the world, because it actually is creating organic compounds, which then are the source of energy for life, out of air, from the carbon dioxide in air, which is a pretty amazing process if you really think about it. And it's definitely the most abundant enzyme. So this is just showing you again the Calvin cycle and what's going into and out of this process. So in the Calvin cycle, we have our carbon dioxide and our ATP and NADPH, which are going in to make, as a product, things like glucose, and then ADP and NADP+. And this is a final summary of photosynthesis. Although we haven't assigned section 10.4 for reading, I want to go over just a couple things that you should know about alternative mechanisms for fixing carbon in the Calvin cycle. And these are believed to have evolved due to hot, arid conditions, so that plants can actually go through photosynthesis without losing too much of the materials that are needed for photosynthesis to occur. So dehydration is a problem for plants who are trying to survive in hot, arid climates. And so what is required is a trade-off with the efficient metabolism that's occurring through photosynthesis. So on hot, dry days, plants are naturally going to close their stomata, those little pores on the bottom of their leaves, in order to keep the evaporation or transpiration of water from the leaves from being too great a loss. But when we have those stomata closed, the other thing that cannot pass into the leaves is carbon dioxide. And so photosynthesis is limited for that reason. It also keeps oxygen, the O2 that is being produced by photosynthesis and the splitting of water. The closing of the stomata is going to keep the O2 from being released from the plant, and so it's going to build up. So what happens is the plant goes into a process called photorespiration. And there are two types of plants that use photorespiration. There's a C4 plant and the CAM plant.